DNA replication. So I really hope you guys understand it well. So isn't it amazing how powerful Allah Subhanahu Wa Taala is? Iza, can you try to read Surah Al-Mu'minun verse 14 until 15?
Yes, Doctor. أعوذ بالله من الشيطان الرجيم ثم خلقنا النطفة علقة فخلقنا العلقة مضغة فخلقنا المضغة عظاما فكسونا العظام لحما ثم أنشأناه خلقا آخر فتبارك الله أحسن الخالقين ثم إنكم بعد ذلك لميتون صدق الله العظيم. Yang bermaksud, kemudian air mani itu kami jadikan sesuatu yang melekat. Lalu sesuatu yang melekat itu kami jadikan segumpal daging. Dan segumpal daging itu kami jadikan tulang belulang. Lalu tulang belulang itu kami bungkus dengan daging. Kemudian kami menjadikannya makhluk yang berbentuk lain. Maha suci Allah, pencipta yang paling baik. Kemudian setelah itu, sungguh kamu pasti mati.
Alright, thank you Iza. So guys, that's all for today. See you guys next week. Bye bye.
Thank you, Doctor.
I don't understand a single thing on the topic that we just learned. What about you? Did you get anything from the lecture?
I think we are on the same page. I'm planning to meet Dr. Nis after class to ask for further explanation about DNA replication. Do you want to join?
Yes, please. Doctor. Sorry, doctor, but can you further explain about DNA replication, please?
Sure.
So what is DNA replication? DNA replication is a process of producing two identical copies of DNA from one original DNA molecule.
So, DNA replication only occur during S phase in interphase and in semi-conservative model.
In semi-conservative model, two parental DNA separate. Each of them will act as the template. A new complementary strand of DNA will be synthesized from the template.
After first replication, two new DNA molecules will produce. Each consists of one parental strand and one newly synthesized strand.
So, these are the enzymes that are involved in DNA replication. Let's go.
Wait. Do you think that's the only enzyme involved in DNA replication? Well, there's actually two more left with another protein.
Next, moving on to mechanism of DNA replication. There are 6 mechanisms in DNA replication. So, we're going to delve into it deeper now.
The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. Next, the separated strand each provide a template for creating a new strand of DNA. An enzyme called primase then start the process. This enzyme made a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA.
An enzyme called DNA polymerase binds to the primer and will make a new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' to end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made and the process is repeated again. Once the new DNA has been made, the enzyme isonuclease removes all the RNA primers from both strands of DNA.
Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
فبأي آلاء ربكما تكذبان